All right, so last video is kind of all over the place, just going through all of the different parts and steps of muscle contraction. So this picture right here, I think really breaks it down. So follow along. I'm going to start at the top left there. I have my axon terminal that is part of the neuron, and it has these things called synaptic vesicles. These synaptic vesicles are going to release acetylcholine, which travels across the synaptic cleft. That acetylcholine bonds to the motor end plate at these things called acetylcholine receptor sites. Once they do that, we've plugged in the lamp. Now our muscle cell or muscle fiber is starting to get electricity or an action potential or excitation. If you follow the arrows to the right, that action potential is going to travel along the sarcolemma, which is, again, the cell membrane of the muscle cell, and then go into the T-tubules. The T-tubules, you follow the arrows down, you notice those are, that's taking this excitation deep into the muscle cell. Now, if you look at on this picture, you see these two, I guess, purplish, bluish things on the outside of the T-tubule. Those are the cisternae. Those are just a connection to the sarcoplasmic reticulum. So now that signal has gone from the T-tubule and it's been passed to the cisternae, and then that cisternae transfers that excitation to the sarcoplasmic reticulum. Once we have that, the sarcoplasmic reticulum starts releasing positive calcium ions into the sarcolemma, and that calcium goes down and will bond to the troponin on the actin molecule. Once that bond happens, so now I am down there at step 3, it bonds to the troponin. That's going to shift the tropomyosin, and that allows the myosin heads to attach, forming our cross bridges. Once those attach, our myosin cross bridges are going to basically bend and go through this thing called the power stroke and pull the actin towards the center of the sarcomere. And it does that over and over and over again. So if you've ever played tug of war, you grab onto the rope, you pull, and then you grab on farther down, and you pull again, and you pull again. You do that until you win tug of war. Well, that's basically what the myosin heads are doing. They attach, they pull, and then they detach, attach farther down, pull, so on and so forth. So that's what allows us to contract our muscle. That whole thing's called the sliding filament model. Remember, it's sliding filament because these filaments slide past one another. They are not changing shape. When we're done contracting, okay, now I'm down to step number 5. The calcium gets pumped back into the sarcoplasmic reticulum. Now, it does this because if up at the synapse, the synaptic cleft and the motor end plate, the acetylcholine is being broken down by acetylcholinesterase. And then finally, after that calcium's gone, if you look at step 6, it says the tropomyosin blockage is restored. So we are blocking those sites so the cross bridges cannot attach anymore, and we've relaxed our muscle. So that's the whole process. Seems really short and easy when I talk about it on one slide, but it kind of involves every step that we've gone through all the way from the top of the muscle.